Hi, my name is Demi. I'm a PhD candidate at the Australian National University, and I'm here at the CSIRO, or the Australian Cotton Research Institute in Narrabri, New South Wales. Here at the Australian Cotton Research Institute, we specialise in developing and researching Australian cotton cultivars that are developed for Australian conditions. Some of our primary traits that we try to improve include photosynthesis and water use efficiency. Improving these traits requires an understanding of how the sugars from photosynthesis and the water from the soil moves around the cotton plant. Both of these processes rely on an understanding of vascular tissue, and that's what we're going to be discussing in today's video.
There are three major tissue types in plants, all with their own functions important for the plant's growth, development, and survival. Firstly, dermal tissue forms a barrier to the environment and includes the outer layers referred to as the epidermis, stomata which control the flux of gases and vapors into and out of the plant, and surface hairs called trichomes. Secondly, ground tissues include specialized cells such as photosynthetic parenchyma or structural sclerenchyma. Finally, vascular tissue, similar in nature to the circulatory system in animals, is required for transportation of water, nutrients, and other important molecules throughout the plant. Vascular tissues form long, vein-like cells that form a transportation network throughout the plant, much like the veins and arteries in animals. They form a continuous connection from the root tip in the soil to the surface of the leaf, spanning the entire length of the plant. There are two primary types of vascular cells called xylem and phloem. These clump together to form vascular bundles, which vary in their size and arrangement depending on the organ of the plant. For example, the arrangement of vascular bundles in a root look remarkably dissimilar to those of a stem or a leaf, because the transportation requirements of these organs is very different. Xylem cells are dead cells, having undergone programmed cell death as they mature after cell division to form a continuous network of hollow xylem. There are two types of xylem cells, called tracheids and vessels. Tracheids are smaller in both diameter and length, and they taper at each end where they connect with the next tracheid cell. Pits between tracheid cells enable passage of water between these narrow cells. Vessels are larger in diameter and length, forming a stacked column of hollow cells, much like a straw. Water is transported passively through xylem cells in a continuous column according to the cohesion tension theory, which relies on cohesion of water molecules to one another to pull the unbroken column of water up the xylem. As water is evaporated through transpiration through stomata at the leaf surface, the water is pulled in a continuous column through the xylem, dragging the water column up the length of the plant. This process generates high negative pressure within the xylem cells. Therefore, xylem cells are reinforced with lignin for strength and structure. In drought situations, higher pressure or water tension can cause the water column to break, which incorporates air into the xylem. This is referred to as an embolism or cavitation and restricts further movement of the water through the xylem, worsening the ability of the plant to remain hydrated. The phloem is comprised of a column of hollow living cells called sieve tube elements, or simply sieve elements, which are accompanied by a companion cell. The sieve elements are connected by perforated walls to form a channel for the movement of sugars and other molecules such as amino acids and minerals. The phloem sap can move both up and down the plant, unlike unidirectional xylem. Think of the phloem like a highway. Its primary role is to transport the goods, in this case sugars, from the factories, or the leaves, to the consumers or grocers, which in this case are the other parts of the plant where they are required. Transport through the phloem is complex, but it is generally considered to be an active mechanism driven by an osmotically generated pressure gradient. This process is described as mass phloem loading. As products such as sugars build up around the source or the leaves, water molecules are drawn into the phloem from the xylem to these regions of high solute concentrations. As the pressure builds up from the entering water, the phloem increases in pressure, allowing the contents to be pushed down the pressure gradient. The sugars and other nutrients are released or unloaded at their sinks, for example the roots or developing flower. The unloading process can be active, such as through the use of sugar transporters, or passive via diffusion.